We first gathered 3 organic solvents in deionized water and grouped them into beakers and covered them with cellophane. We then cut 4 TLC plates and drew a pencil line about 1 centimeter from the bottom and spotted each plate with our dyes and the unknown. We then put each TLC plate in each kind of solvent and allowed the solvent line to rise. After it rose a fair amount, we took each TLC plate out of the solvent and measured the distance traveled by each dye. We used these measurements to calculate the RF of each dye and our unknown and determined that the glacial acid mixture gave the best separation. We then began to assemble our column by plugging the Pasteur pipette with a small amount of glass wool. Next, we added sand on top of the glass wool and then filled the Pasteur pipette with silica gel until it was about half an inch from the top. We then saturated the column with the glacial acid mixture and made sure the solvent stayed just above the silica gel. We immediately added 2 drops of the unknown and continued to add a little bit of solvent to ensure that the unknown could flow through the column. We covered this with a little bit of sand and then we allowed the solvent to drip through. We collected the clear solvent in a waste beaker and then interchanged the beakers when we noticed that color began to drip through. This would ensure that each dye in the unknown were collected separately. We placed a small portion of the collected dyes in cuvettes, diluted them, and measured their absorbance from 200 to 800 nanometers and compared them with known values.